right, so get ready for another deep dive. This time, it's into the fascinating world of cell biology.
Oh, this is going to be fun.
We've got a stack of biology textbooks and articles here, and you know what else we have? Some incredible stories about Canadian scientists.
I love those.
It's going to be like our own personalized biology class,
Cool.
like shrinking down to microscopic size.
To explore the amazing world inside a cell.
Exactly.
It's mind-blowing when you think about it. Everything we see, touch, experience, all built from these tiny building blocks.
Okay, so let's break it down. We all know living things are made of cells.
Right.
But what are those cells actually made of?
Well, imagine you're building with Legos.
Okay, I like where this is going.
You need all sorts of different shapes and sizes to make something complex, right?
Of course.
Cells are similar, but they're built from these incredibly small molecules.
So like nano Legos.
Exactly. Think water, oxygen, and carbon dioxide.
Okay, so water. I mean, I know that's H2O,
Yeah.
but why is it so important for cells?
Water is like, I don't know, it's the lifeblood of a cell.
Wow.
It's a fantastic solvent, meaning it can dissolve tons of substances, which is essential for, moving nutrients and waste within a cell.
Okay, so it's like a transportation system.
Pretty much. Plus, it helps regulate temperature.
Oh, that's important.
And get this, it actually expands when it freezes.
Really?
Yeah.
Yeah.
That's crucial for aquatic life. All these amazing abilities come from its unique structure.
Wow, so water is like the ultimate multitasker. What about oxygen and carbon dioxide?
Well, oxygen is essential for many cells to release energy from food. It's kind of like the fuel that powers a fire.
Makes sense.
Then carbon dioxide is a byproduct of that process, like the smoke from a fire.
I see.
Think about how we breathe in oxygen and breathe out carbon dioxide.
Yeah.
That same exchange happens at the cellular level, too.
That's so cool. So we've got these small essential molecules, but how do they come together to build bigger structures in a cell?
That's where organic compounds come in.
Organic compounds.
Think about the food we eat. Bread, oil, meat. They're all made up of organic compounds, which contain both carbon and hydrogen.
Ah, okay.
But carbon is especially important.
How so?
Well, it can form 4 bonds with other atoms.
4?
Yeah, making it incredibly versatile for building those complex structures.
So like a super flexible Lego piece.
You got it. It's that bonding power of carbon that lets it form chains, rings, complex structures. It forms the foundation of bigger molecules called macromolecules.
Macromolecules. Now we're getting to the good stuff.
Yeah.
So bread, oil, and meat, are those connected to the key macromolecules?
Absolutely. Bread is mostly carbohydrates, oil is made up of lipids, and meat is primarily protein.
Okay.
These macromolecules are like the building blocks of a cell, each with its own unique structure and function.
So it's like using different types of Lego bricks to build different parts of a spaceship.
Exactly. Let's start with carbohydrates. They're like the cell's primary energy source. Think of them as chains of sugar molecules.
Like strings of pearls.
Perfect analogy. Plants store energy as starch, while animals store it as glycogen. Both are complex carbohydrates made up of glucose units.
So when I eat a slice of bread, my body's breaking down those chains for energy.
That's it. And those quick bursts of energy you get, that's your body tapping into those glycogen stores. Now, lipids, things like fats and oils, they're like energy reserves. They store energy long-term and provide insulation, like a warm coat for cells.
Oh, like the blubber on a walrus?
Exactly. And here's a fun fact. A gram of fat stores more than double the energy of a gram of carbs.
Wow, no wonder we crave those fatty foods sometimes.
It's all about survival.
Right, it all makes sense now. But how are fats structured differently from carbs?
Think of fats as 3 fatty acid chains attached to a glycerol backbone, kind of like a 3-pronged fork.
Okay, I can picture that.
Now, these fatty acid chains can be saturated or unsaturated.
And that determines whether a fat is solid or liquid at room temperature, right?
Exactly. Saturated fats, like butter, are solid because their chains are straight and pack tightly together.
But unsaturated fats, like olive oil, are liquid.
Exactly. Their chains have kinks, preventing them from packing as closely.
So that's why butter is solid and oil is liquid. It all comes down to those fatty acid chains. All right, what about proteins? They seem like the most diverse of the bunch.
Proteins are like the multi-tool of the cell.
Really?
They do it all. They act as enzymes to speed up reactions, transport molecules, transmit signals, and provide structural support.
Wow.
They're involved in almost every aspect of cell function.
But how can they be so diverse if they're all made of the same building blocks?
Imagine a necklace made of beads. You have 20 different types of beads representing the 20 different amino acids.
Okay, I'm following.
By changing the order and number of beads, you can create an almost infinite variety of necklaces.
Each with its own unique pattern and function.
Exactly.
So the sequence of amino acids determines the specific structure and function of a protein.
Precisely. And the way these chains of amino acids fold and twist also plays a role.
They form intricate 3D shapes.
Absolutely, much like origami. And that precise shape is critical for their function. Think about a key fitting into a lock. If the shape of the key is even slightly off, it won't work.
Right, it has to be a perfect match.
The same goes for proteins.
So a protein's shape is crucial for its function. But what happens if that shape gets messed up?
Well, if a protein is exposed to extreme temperatures, pH, or harsh chemicals, it can unfold or change shape. We call that process denaturation.
Denaturation.
It's like crumpling up a carefully folded origami crane. It loses its form and can no longer fly.
Got it. So we've talked about the building blocks of a cell, but what about the boundary that separates a cell from its surroundings?
That's where the cell membrane comes in.
The cell membrane.
Think of it as the city walls, controlling what goes in and out.
Okay, I like that analogy.
It's the gatekeeper of the cell, ensuring that the internal environment stays stable and protected.
And I remember reading that the electron microscope was key to understanding its structure. Wasn't there a Canadian connection there?
Absolutely. The first truly usable electron microscope was built right here in Canada at the University of Toronto in 1938.
Wow.
Two brilliant graduate students, James Hillier and Albert Prebus, were behind this groundbreaking invention.
Talk about Canadian ingenuity. So what did this powerful microscope reveal about the cell membrane?
It revealed that the cell membrane is a bilayer,
Bilayer.
a double layer of molecules called phospholipids. Imagine them shaped like lollipops with two sticks.
Okay, I can picture that.
The round head of the lollipop is the phosphate group. It's hydrophilic, meaning it loves water.
It loves water.
But the two sticks are the fatty acid tails, and they are hydrophobic.
Which means they fear water.
You got it.
So the cell membrane is like a double layer of lollipops with their water-loving heads facing outwards and their water-fearing tails tucked in between.
That's a great analogy. We call it the phospholipid bilayer, and it's the foundation of the cell membrane. But it's not just a static wall. It's dynamic and fluid with proteins and other molecules embedded within it, constantly moving like a mosaic. We call it the fluid mosaic membrane model.
Wow, so the cell membrane is like a constantly moving mosaic of molecules. That's incredible. And it's also selectively permeable, right? Not just anything can pass through.
Exactly. It acts like a border crossing, carefully controlling what enters and exits the cell, using a variety of mechanisms to do so.
Okay, this is where it gets really interesting for me. How does a cell actually transport substances across its membrane?
Think of it as different modes of transportation. Some molecules can simply diffuse across the membrane,
Diffuse.
moving from an area of high concentration to low concentration, like passengers walking through an open doorway.
That's like how the smell of freshly baked cookies spreads through a house. The cookie molecules are moving from a high concentration in the kitchen to a lower concentration in other rooms.
Perfect analogy. We call that process diffusion. Water also moves across the membrane through diffusion, but we give that a special name, osmosis.
Osmosis.
It's the movement of water from a region of high water concentration to a region of low water concentration.
So osmosis is just diffusion, but specifically for water molecules.
Yeah.
What determines which way water moves during osmosis?
It's all about balance. Water will always try to move to the side with a higher concentration of dissolved substances to even things out. Imagine two rooms connected by a door, one packed with people and the other mostly empty.
Okay.
People would naturally move from the crowded room to the less crowded one until both rooms have a similar number of people.
That makes sense. So if a cell is placed in a solution with a higher concentration of solutes than inside the cell, water will move out of the cell, causing it to shrink.
Exactly.
And if it's in a solution with a lower concentration of solutes, water will move into the cell, causing it to swell.
You got it. We use the terms hypertonic, hypotonic, and isotonic to describe these different solutions. Hypertonic solutions have a higher solute concentration. Hypertonic solutions have a lower solute concentration. And isotonic solutions have an equal solute concentration.
Got it. Hypertonic means more solutes, hypotonic means fewer solutes, and isotonic means equal solutes. So diffusion and osmosis are passive processes. They don't require the cell to expend energy. But what about when a cell needs to transport larger molecules or move substances against their concentration gradient? It's like trying to swim upstream.
Right. That's when the cell needs to be a bit more proactive and use energy. One way it does this is through facilitated diffusion.
Facilitated diffusion. So it's still diffusion, but the cell gives it a little help.
Exactly. Special proteins in the cell membrane act like channels or carriers, helping larger molecules or charged particles cross the membrane.
Okay.
It's still a passive process, though, because the molecules are moving down their concentration gradient, like floating downstream with a little help from a friendly current.
Okay, so facilitated diffusion is like providing a special lane for certain molecules to cross the border. But they're still moving in the direction of lower concentration. What about when the cell needs to move molecules uphill against their concentration gradient?
That's where active transport comes in. It's like pushing a boulder uphill. It requires energy. The
Wow.
cell uses specialized transport proteins that act like pumps,
Bump.
using energy from ATP to move molecules from a region of low concentration to a region of high concentration.
So active transport is like hiring a team of movers to carry that boulder uphill.
Exactly.
And those movers need energy to do their job. What's a good example of active transport in action?
The sodium-potassium pump is a prime example.
Okay.
It's constantly working to maintain a higher concentration of potassium ions inside the cell and a higher concentration of sodium ions outside the cell.
Right.
This gradient is essential for nerve impulses and muscle contractions.
Wow. So active transport not only moves molecules, but it also sets up these essential gradients that drive other cellular processes.
Amazing.
Okay, so we've covered diffusion, osmosis, facilitated diffusion, and active transport. Are there any other transport mechanisms we need to know about?
We've covered how cells transport small molecules across their membranes, but what happens when they need to move really big things, things that are too large for those other transport mechanisms?
Good question. It's like trying to fit a grand piano through a standard doorway.
Exactly. You need a different strategy. In those cases, cells use bulk transport mechanisms.
Oh?
They can engulf substances by forming vesicles,
Vesicles.
little membrane-bound sacs, in a process called endocytosis. It's like the cell eating or drinking.
Cell eating and drinking. I love these analogies. Can you break down the different types of endocytosis?
Sure. There's penocytosis, which is like cell drinking, where the cell takes in small droplets of extracellular fluid. Then there's phagocytosis, which is like cell eating, where the cell engulfs larger particles like bacteria or cellular debris.
Got it.
And finally, there's receptor-mediated endocytosis, where specific molecules bind to receptors on the cell surface,
Okay.
triggering the formation of a vesicle to bring those molecules inside.
Ah, so it's like a lock and key mechanism. Only the right molecule can bind to the specific receptor, triggering the uptake process. I remember reading a fascinating story about a Canadian scientist, Dr. Kenneth Story, and his research on frozen frogs. It mentioned something about a natural cryoprotectant. Is that connected to endocytosis?
It is. Dr. Story's research focuses on how some frog species survive freezing solid during the winter.
Wow, freezing solid.
He discovered that these frogs flood their cells with glucose,
Oh, wow.
acting as a natural antifreeze, preventing damaging ice crystals from forming. It's possible that this glucose uptake is facilitated by endocytosis.
That's incredible. So these frogs have evolved a way to use endocytosis to protect themselves from freezing. Imagine the implications that research could have for organ preservation and transplantation in humans. That's mind-blowing.
It's a fascinating area of research with potential implications for the future of medicine.
Yeah.
Now, we've talked about how cells bring things in through endocytosis, but how do they get rid of waste or secrete molecules?
It's like taking out the trash, right?
Exactly. It's called exocytosis, and it's essentially the reverse of endocytosis. Vesicles inside the cell fuse with the cell membrane, releasing their contents outside the cell.
Okay.
It's how cells get rid of waste products, secrete hormones, or even send signals to other cells.
So endocytosis is like bringing groceries into the house, and exocytosis is like taking out the trash. It's amazing how cells have these intricate systems for moving things in and out. Now, we've been talking about a generic cell, but in multicellular organisms, not all cells are created equal, are they?
You're absolutely right. Multicellular organisms have a division of labor among their cells, much like a well-organized society.
Oh, I see.
Different cell types are specialized for specific functions, and they work together in a coordinated manner to form tissues, organs, and systems.
So it's like a cellular society with different professions, muscle cells for movement, nerve cells for communication, blood cells for transport, and so on. It's mind-boggling to think about how all these specialized cells work together.
It truly is, and each of these specialized cells has unique structural features that allow it to perform its specific function efficiently. We'll delve deeper into these specialized cells and their fascinating adaptations next time.
That's a great cliffhanger. I can't wait to learn more about these cellular specialists and how they contribute to the overall symphony of life.
Yeah.
So last time, we talked about the amazing world of cells, their structure, how they transport things.
Right.
But we didn't get into the specifics of how they create those complex molecules, the proteins.
Right.
It's like learning all the ingredients in a recipe, but not how to actually bake the cake.
That's a great analogy. Making proteins is a lot like baking a cake. A multi-step process, but really fascinating.
Okay, so walk me through it. Where does the recipe for making proteins come from?
The recipe is stored in the cell's DNA. It's like the master blueprint for the entire cell.
A massive cookbook.
Yeah. Each gene within the DNA, that's a specific recipe for building one protein.
So each gene is like a recipe for one protein. Okay, got it. But how does the cell actually read and follow those recipes?
It's a two-step process.
2 steps. Okay.
It starts with transcription.
Transcription.
Imagine you want to bake a cake,
Okay.
but you don't want to take the whole cookbook into the kitchen.
Right.
You'd copy down the recipe, right?
Of course.
That's essentially what happens during transcription. The cell creates a messenger molecule.
What's it called?
It's called messenger RNA, or mRNA. It's basically a copy of the gene's instructions.
So the mRNA is like a temporary copy of the recipe,
Yeah,
carrying it from the DNA in the nucleus out to the cytoplasm where the real action happens.
exactly.
Okay, what happens next?
The mRNA travels to a ribosome. That's like the chef in our baking analogy.
Okay.
Ribosomes are the sites of protein synthesis, where those instructions are put into action.
Ribosomes, those tiny 2-part structures, right?
Yeah.
Found throughout the cytoplasm. I remember seeing images of them. They kind of look like little hamburger buns.
That's a great way to visualize them. At the ribosome, the mRNA is, read and translated into a chain of amino acids.
So we call that process translation.
Right. It's like translating the recipe from the language of nucleic acids into the language of proteins.
Translation, I get it. But how does the ribosome know which amino acids to link together and in what order?
That's where another type of RNA comes in. Transfer RNA, or tRNA.
tRNA.
Each tRNA molecule carries a specific amino acid and has a 3-nucleotide sequence called an anticodon.
Anticodon.
Think of it like a code word that matches a specific codon on the mRNA.
So it's like a matching game. Each tRNA molecule carrying the right amino acid and its corresponding code word, ensuring that the amino acids are linked together in the correct order based on those mRNA instructions. Pretty clever.
It's incredibly intricate, a super precise process. As the ribosome moves along the mRNA, it matches the codons with the anticodons on the tRNA molecules, linking the amino acids together one by one, like assembling a chain of beads.
So once the ribosome reaches the end of the mRNA, we have a complete chain of amino acids, which folds into that specific 3D structure of the protein.
Exactly. It's remarkable. From DNA to RNA to protein, a beautifully choreographed dance of molecules. And remember those cellular recycling centers, the lysosomes?
Lysosomes.
They play a crucial role in keeping this whole process running smoothly.
Lysosomes, they're like the cell's waste disposal and recycling units. How do they actually work?
Imagine them as tiny stomachs within the cell, filled with powerful digestive enzymes. They can break down a wide range of molecules, from worn-out cell parts to substances brought in through endocytosis, like a cellular cleanup crew, making sure everything stays tidy and efficient.
So if a cell has a damaged mitochondrion or some cellular debris, it sends it to the lysosome to be broken down.
Precisely. The lysosome fuses with the vesicle containing the unwanted material, and those enzymes get to work, breaking it down into smaller molecules, which can be reused or discarded.
It's like a cellular shredder, breaking down those unwanted components. But what happens to those smaller molecules once they're broken down? Does the cell just get rid of them?
Not necessarily. The lysosome isn't just a disposal unit, it's also a recycling center.
Oh, cool.
Those smaller molecules are transported back into the cytoplasm, where they can be reused to build new molecules or provide energy.
So the lysosome's like a zero-waste facility, breaking down old components and repurposing the materials to create new ones. It's incredibly efficient.
It is. And this constant cycle of breaking down and rebuilding is crucial for maintaining cellular health and function.
This is all starting to make sense. We've talked about how cells are organized into tissues, organs, and systems, each with specialized functions. It's like a well-oiled machine. with each part playing a vital role.
And the efficiency of this machine depends on the coordinated efforts of all those specialized cells.
Right. We discussed examples of specialized cells like, heart muscle cells, parietal cells in the stomach, podocytes in the kidneys, and squamous epithelial cells in the lungs. They're all so different, but they work together seamlessly.
They do. It's truly remarkable how diverse cells can be while still working together harmoniously. But sometimes things can go wrong. Cells become damaged or dysfunctional, leading to various diseases.
Oh, right.
Yeah.
That's a sobering thought. We talked about diabetes, which affects how cells use glucose. What are some other examples of diseases that disrupt cellular function?
Well, cancer is a prime example. It's a disease of uncontrolled cell growth and division.
Uncontrolled.
Yeah, cells usually have a carefully regulated cycle of growth and division.
Right.
But in cancer, these controls break down.
So it's like the cell's growth control mechanisms have gone haywire. But how does that happen at the cellular level?
It's complex, but it often involves mutations in genes that control cell growth and division. These mutations can be caused by a variety of factors, like exposure to carcinogens, radiation, or just errors during DNA replication.
So these mutations disrupt the normal signaling pathways that regulate cell growth, leading to uncontrolled proliferation and the formation of tumors. What are some of the ways researchers are trying to combat cancer?
One promising approach is immunotherapy. It harnesses the power of the immune system to target and destroy those cancer cells. Another is targeted therapy, using drugs that specifically target molecules involved in cancer cell growth.
So it spares the healthy cells.
Yeah.
Targeted therapy sounds like a more precise approach, minimizing damage to healthy tissues. What are some other areas of research in cell biology that you find particularly exciting?
The field of regenerative medicine holds immense promise.
Oh, yeah.
Researchers are exploring ways to use stem cells to repair damaged tissues and organs, potentially offering new treatments for a wide range of diseases, from heart disease to spinal cord injuries.
That's incredible. It's like harnessing the power of cells to rebuild and regenerate damaged tissues. It really highlights the potential of cell biology to improve human health.
It does. And as our understanding of cells continues to grow, so too will our ability to develop new and innovative treatments for diseases that currently have no cure. We're just scratching the surface of what's possible.
Well, this deep dive into the world of cells has been incredibly eye-opening. From protein synthesis and lysosomal recycling to specialized cell types and the complexities of disease, we've covered so much ground.
We have, and there's still so much more to explore. But for now, let's take a break and digest all this amazing information. We'll continue our journey into the cellular world soon.
So we've spent this whole deep dive exploring, the inner workings of a single cell.
Right.
From its basic building blocks to these intricate processes.
Yes.
But now I'm kind of curious. How do all these individual cells communicate with each other to form tissues, organs, and ultimately a whole organism? It can't just be random chaos, can it?
You're right, it's definitely not random. Cells are incredibly organized, and they communicate with each other constantly. Yeah, much like individuals in a bustling city.
Wow.
They use a variety of methods, including chemical signals, electrical impulses, even direct contact to share information, coordinate their activities. It's like this complex communication network keeping everything running smoothly.
Okay, that makes sense. So what are some examples of these cellular communication methods?
Well, hormones are a classic example of chemical signaling.
Hormones.
They act like long-distance messengers,
Okay.
traveling through the bloodstream to target cells in different parts of the body.
So think about how adrenaline surges through your body in a stressful situation, preparing you for fight or flight.
Exactly.
That's hormone-mediated communication in action.
Yes.
Or how insulin, produced by the pancreas, travels to cells throughout the body to regulate those blood sugar levels.
Right.
What other chemical signals do cells use?
Neurotransmitters are another important example. They're released by nerve cells at synapses,
Synapses.
those tiny gaps between nerve cells, to transmit signals to other nerve cells, muscle cells, or gland cells.
Okay.
It's how our brains communicate with the rest of our bodies,
Oh, wow.
controlling everything from muscle movements to our senses.
So those are the signals that allow us to react quickly to our surroundings, like when we touch something hot and instantly pull our hand away.
Exactly.
That's incredible.
Yeah.
What about electrical impulses? How do cells use those to communicate?
Nerve cells are the champions of electrical signaling.
Okay.
They generate electrical impulses called action potential
Action potentials.
that travel along their long, thin axons. transmitting information rapidly over those long distances. It's like sending a message via telegraph, but at lightning speed.
So it's like an electrical circuit, with nerve cells acting as the wires transmitting signals throughout the body. How do these electrical impulses actually work?
It involves the movement of ions across the cell membrane. Remember the sodium-potassium pump?
The pump, yeah.
Which maintains that crucial concentration gradient of sodium and potassium ions. That gradient is essential for generating these electrical impulses,
Oh, wow.
allowing nerve cells to fire signals rapidly and efficiently.
So the pump isn't just moving molecules.
Right.
It's also setting the stage for these electrical conversations to happen.
Exactly.
Fascinating. Now, what about direct contact between cells? How do they communicate that way?
Some cells have specialized junctions
Okay.
that allow for direct communication with their neighbors. One example is gap junctions.
Gap junctions.
Tiny channels that directly connect the cytoplasm of adjacent cells,
Yeah.
allowing small molecules and ions to flow freely between them. It's like having a secret passageway between houses.
So it's like a direct pipeline between cells, allowing for rapid and synchronized communication. I remember reading that plant cells have something similar called plasmodesmata.
That's right. Plasmodesmata are channels that connect the cytoplasm of adjacent plant cells, facilitating the transport of molecules and communication between those cells.
Okay.
Think of it as a network of tunnels.
Connecting the cells throughout a plant.
Yeah, allowing them to share nutrients, water, and signals.
It's amazing how cells have evolved these diverse and sophisticated mechanisms for communicating with each other. It's like a symphony of signals, coordinating all those intricate cellular processes that keep us alive and functioning.
It truly is. It highlights the fact that cells are not isolated entities. They're part of a larger community, working together to maintain the health and harmony of the entire organism. Just like in any society, communication is key.
This has been an incredible journey into the world of cells. From their basic building blocks to their complex communication networks, we've explored their structure, function, and their role in creating this amazing tapestry of life.
And there's still so much more to discover. But for now, let's savor the knowledge we've gained and appreciate this remarkable world that exists within every single cell of our bodies.
Thank you for joining us on this deep dive into the fascinating world of cell biology. Until next time, stay curious.